you very much. So thank you for asking me to speak about the basics of cilia in PCD. I'm going to start off with what are cilia. So cilia are tiny little projections that line the airways, and they're very small. I've got some facts on the side here. So the word cilia comes from the Latin word for eyelash because they're tiny little hairs. We have thousands of cells in our body, and that for every cell there's 200 cilia. So you can't see them with the naked eye. They're less than 1,000th of a centimeter in size, and they beat and move about 15 times each second. So they're moving quite fast. Their job is as the first defense of our body and our lungs against things that we inhale. So protecting against particles, viruses, bacteria, which get stuck in the mucus of the airways, and then the cilia waft and beat them out. So they're like an escalator from our lungs to keep our lungs clean and clear. As I said, they're very small, and the first person that described cilia was a man called Anthony van Leeuwenhoek, and he essentially was one of the first people to have a microscope. So you need a microscope to be able to see the cilia outside the body. And when we're doing diagnosis for primary ciliary dyskinesia, what we do is we take a sample from the nose, which is where the cilia start. They start in the nose and they line our entire airways. We take a sample from the nose and then we look at that under a microscope. And there's just a picture here of one of the microscopes that we have in our center, just so you can see what that looks like. It's got a big chamber that keeps things at 37 degrees, so it keeps things at body temperature, and lenses that allow us to see the cilia. This top left-hand video is a video looking through that microscope at cilia beating. As I said, they move about 15 times per second, so this is actually slowed down. So it's a slowed down movie, and you can see that the cilia beat in a coordinated way, and they try to clear things out of the airways. People with primary ciliary dyskinesia, cilia don't beat in the same way or properly, but because PCD is quite different depending on what gene you have, actually the way the cilia are ineffective is quite different. So I've just put some examples on here. This top right one is, I think, what we traditionally think of as PCD, which is completely static immotile cilia that aren't moving at all. But we also see, when we're doing PCD diagnosis, cilia that only the top half is moving and not the bottom half. So you can see there's still some movement, but it's not effective at clearing things out of the airways. And then this bottom right-hand corner, hopefully that's showing well. You can also see these cilia have big bulbs on them and are moving in all sorts of different directions and are also ineffective. So not everybody is the same in how their cilia are affected by PCD. If you look at one of those tiny little hairs and you look much closer using a bigger microscope, so this is an example of an electron microscope here with Claire, who's going to talk later on, and me from a few years ago, but you can see it's taller than a person. So this is the electron microscope, and if you take one of those tiny little hairs and you slice it in half, I always think of it being a bit like bits of rock you get at the seaside. If you slice it in half and look at it in the center, then this is what you get. So this is very, very small now, and the important parts of this are these tiny little hooks, which are called the dynein arms. And those are the motor proteins that can make the cilia beat. Oh, actually, I've colored them in. I did a better job than I remembered with my slides. So the purple is the outer dynein arm, which is the motor protein that makes the cilia move. And again, like the beating pattern is different in PCD for different people, also the things that you see in the structure is different. So many people with PCD have these missing motor proteins that make the cilia beat. So they have missing outer dining arms. And C here is an example of that. So you can see there's a gap here where that motor protein should be. Some people have missing outer dining arms and inner dining arms, which is the other set of motor proteins. And in some cases, the radial spokes are affected, and these are like the spokes of a wheel. And you can imagine when they're not there, or when the bits that link these microtubules, as they're called, aren't there, then the structure collapses. And in some people with PCD, we actually can't see, even looking at this magnification, why the cilia aren't beating properly. So we can see on the videos that they're not being effective, but even looking at this structure, we're unable to see why that is. And that's where genetics comes in. So whereabouts in our body are cilia? Myrona's going to talk a lot, I think, after me about symptoms. And I think if you think about where cilia are in the body, then a lot of symptoms of PCD make sense. So cilia start in our nose and they line our airways right down to the very small airways to keep the airways clean. They also line our ears. And their job there is in fluid in the ears. During development, when babies are, or when the embryo is developing, the cilia have a role in determining what is left and what is right. And that is why we get situs inversus sometimes with PCD and reversal of the organs. They're very similar in structure to sperm tails, and also they line the fallopian tubes. There's also a part of the male reproductive system called the efferent duct, and they also line that. And then there's also cilia in the brain that move fluid in the brain. I've lost my genetics slide. Sorry, I skipped over genetics. So we know there's lots of different genes that that cause PCD, and this slide is just an overview of all of those genes. So anything in red is a PCD gene. So some are involved in the growth of the cilia from the cell, others in whereabouts the cilium forms, some in moving things up and down the cilium, and then some in part of this structure itself. And we now know about over 50. So this is a busy slide with lots of information, but the take-home message is there's lots of different genes and they have lots of different functions, but they're all involved in cilia. So I'm going to hand over back to the chairs, and I'm very happy to take questions. Thank you.